The overall goal of this procedure is to test proteins that bind to calmodulin in a calcium-dependent manner. This is accomplished by first collecting proteins of interest. Next, Calmodulin-Sepharose beads are prepared for protein binding. Then, the proteins are incubated with Calmodulin-Sepharose beads. Finally, the proteins that bind to calmodulin in a calcium-dependent manner are eluted. Ultimately, results can be obtained that show proteins that bind to calmodulin in either the presence or absence of calcium through Western blot analysis.
The main advantage of this technique over other existing methods, such as calm chromatography and immunoprecipitation, is that it requires less protein and less time.
This method can help answer key questions in the calmodulin signaling field, such as what proteins bind to calmodulin in a calcium-dependent manner, and how post-translational modifications can affect their binding to calmodulin.
Visual demonstration of this method is critical, as the bead handling steps are essential, and the description of the resuspension and retention of the beads is difficult to follow without seeing it.
To prepare tissue for homogenates, inject organotypic hippocampal slices with a virus containing a plasmid expressing the recombinant protein of interest. In this example, the recombinant protein is GFP-tagged neurogranin. Allow the tissue to express the protein overnight. Approximately 12 to 18 hours after viral injection, add 1 milliliter of dissection buffer to a Petri dish. Transfer the cultured tissue with insert to the Petri dish and add 2 milliliters of dissection buffer to the insert to submerge the tissue. Using a